Review: Taurine, a very essential amino acid. Abstract: Taurine is an organic osmolyte involved in cell volume regulation, and provides a substrate for the formation of bile salts. It plays a role in the modulation of intracellular free calcium concentration, and although it is one of the few amino acids not incorporated into proteins, taurine is one of the most abundant amino acids in the brain, retina, muscle tissue, and organs throughout the body. Taurine serves a wide variety of functions in the central nervous system, from development to cytoprotection, and taurine deficiency is associated with cardiomyopathy, renal dysfunction, developmental abnormalities, and severe damage to retinal neurons. All ocular tissues contain taurine, and quantitative analysis of ocular tissue extracts of the rat eye revealed that taurine was the most abundant amino acid in the retina, vitreous, lens, cornea, iris, and ciliary body. In the retina, taurine is critical for photoreceptor development and acts as a cytoprotectant against stress-related neuronal damage and other pathological conditions. Despite its many functional properties, however, the cellular and biochemical mechanisms mediating the actions of taurine are not fully known. Nevertheless, considering its broad distribution, its many cytoprotective attributes, and its functional significance in cell development, nutrition, and survival, taurine is undoubtedly one of the most essential substances in the body. Interestingly, taurine satisfies many of the criteria considered essential for inclusion in the inventory of neurotransmitters, but evidence of a taurine-specific receptor has yet to be identified in the vertebrate nervous system. In this report, we present a broad overview of the functional properties of taurine, some of the consequences of taurine deficiency, and the results of studies in animal models suggesting that taurine may play a therapeutic role in the management of epilepsy and diabetes. Introduction. The impetus for this review dates back more than a few decades, having originated with a curious malady, i.e., the severe headaches that were often suffered by diners who had ingested monosodium glutamate, a common food additive in general use in homes and restaurants. It came to be known by a variety of names, the most common being the Chinese restaurant syndrome because of its perhaps excessive use in wonton soup. The cause remained a mystery until 1969, when John Olney and his colleagues unequivocally demonstrated the neurotoxic effects of monosodium glutamate. In an impressive series of papers, they showed that when applied topically or by injection, glutamate and its analogues, aspartate, kainate, and methyl D-aspartate, NMDA, alpha-amino-3-hydroxy-5-methylphoisoxazole-propionic acid, AMPA, were cytotoxic to nerve cells in every part of the central nervous system, CNS, 1 to 3. The issue is of more than academic interest, since glutamate-triggered neuronal damage is known to occur when the glutamate concentration of interstitial fluids reaches abnormally high levels as a result of hypoxia, ischemia, or brain trauma. A striking curiosity was seen when Olney's studies were extended to the visual system. In the neonatal mouse retina, for example, he reported that a 30-minute exposure to parenterally administered glutamate, 1 mm, produced a histopathological lesion characterized by swollen cell bodies in the ganglion cell layer, the proximal half of the inner nuclear layer, and extending to the inner plexiform layer. Even after washing and transferring the excised retina to glutamate-free medium, Olney found that the lesion had progressed further, particularly in cells within the inner half of the inner nuclear layer, too. It is noteworthy that although the retina had been bathed in glutamate, only the inner layers were seriously affected. Why had the nerve cells in the distal layers been spared? Neurons and glia have been shown to sequester glutamate via high-affinity uptake systems. These transport mechanisms, regarded as responsible for clearing L-glutamate from the synaptic cleft, 4, 5, and for terminating the excitatory signal, 6, represent the first step in the recycling of the transmitter through the glutamine cycle, 7, 8. Glutamate uptake undoubtedly plays a cytoprotective role, but it is clearly inadequate to spare the inner retina when exposed to toxic levels of glutamate. Rather, it seems likely that there are one or more endogenous substances that serve to protect the outer retina from the typically severe reaction to glutamate. We suggest that one of the most effective endogenous agents protecting the distal retina from the application of toxic levels of glutamate is the amino acid taurine. Other cytoprotectants. Before considering further some of the biochemical and physiological features of taurine, as well as the broad range of conditions in which taurine has been shown to be beneficial, we must acknowledge that the retina may be exposed to several other survival-promoting agents under normal conditions. Many that have been shown to be effective, e.g., brain-derived neurotrophic factor, BDNF, ciliary neurotrophic factor, CNTF, and basic fibroblast growth factor, BFGF, were identified and extensively investigated by Lavale and co-workers, 9–12. These and other members of the transforming growth factor beta family help to protect retinal neurons from ischemia, free radical formation, light damage, and related forms of neuronal insult. Although levels of some of these factors are upregulated in response to injury, 11–13. These agents, even when applied exogenously, primarily tend to slow the cell death process. Treatment with combinations of antioxidants has also proven to effectively rescue photoreceptors in an animal model, RD1, of retinal degeneration. 14. But here too, the agents were applied exogenously. We suggest that the high concentration of endogenous taurine throughout the retina can better serve the role of neuroprotectant against glutamate-induced excitotoxicity. Some functional properties. A broad-spectrum cytoprotective agent. Taurine, 2-aminoethane sulfonic acid, an organic osmolyte involved in cell volume regulation, provides a substrate for the formation of bile salts, and plays a role in the modulation of intracellular free calcium concentration, 15–16. Taurine is one of the most abundant amino acids in the brain and spinal cord, leukocytes, heart and muscle cells, the retina, and indeed almost every tissue throughout the body. It was first identified and isolated from the bile of the ox, Bos taurus, from which it derives its name, 17–18. The chemical structure of taurine, shown in Figure 1a, reveals that it lacks the carboxyl group typical of other amino acids, but does contain a sulfonate group. The major route for the biosynthesis of taurine, shown in Figure 1b, is from methionine and cysteine via cysteinesulfonic acid decarboxylase, CSD, and typically requires oxidation of hypotaurine to taurine as the final step, 19. Open in a separate window. Figure 1. Structure and formation of taurine. A. The chemical formula of taurine is C2H7NO3SMW equals 125.15. B. This oversimplified diagram shows the main steps in the conversion of L-cysteine to taurine. The enzyme cysteine dioxygenase, CDO, catalyzes the conversion of L-cysteine to cysteine sulfonate, and the oxidation of hypotaurine, 2-aminoethane sulfonate, results in taurine. CSD was initially cloned and identified in the liver as the rate-limiting enzyme in the biosynthesis of taurine, 20, and was later shown to be present in the kidney as well as the brain, where it is localized in glial cells. CSD levels are very low in cats, as well as humans and other primates, but the ingestion of meat and seafood, or taurine supplements, helps to maintain normal tissue concentrations of taurine. As Sinwell and Goradisher, 21, have shown, there is an increased incidence of pediatric problems in children being raised on the totally vegetarian diets of vegan communities. Aside from the retina, every region of the brain that has been tested contains or takes up taurine. This includes the pineal, 22, 23, pons medulla, 24, hypothalamus, 25, striatum, 26, and cerebellum, 27, 28. At each of these sites, there is evidence of taurine's ability to ameliorate certain forms of neuropathology. Because it is one of the few amino acids not used in protein synthesis, taurine is often referred to as a non-essential amino acid, or more generously as a conditionally essential amino acid. Considering its broad distribution, its many cytoprotective attributes, 29, 30, and its functional significance in cell development, nutrition, and survival, 31, 32, these are clearly misnomers. Taurine is undoubtedly one of the most essential substances in the body. Moreover, there is ever-increasing evidence that taurine depletion leads to a wide range of pathological conditions, including severe cardiomyopathy, 33, renal dysfunction, 34, pancreatic beta cell malfunction, 35, and loss of retinal photoreceptors, 36. The close relationship between taurine levels and nutritionally induced degeneration is supported further in that taurine supplementation can inhibit light-induced lipid peroxidation, and thereby protect isolated rod outer segments from photic damage, 37, 38. There is a long list of diseases that are impacted by taurine, although the precise biochemical mechanism of action is often not entirely clear. A case in point is its role in diabetes. Numerous studies have indicated that taurine plays a significant role in overcoming insulin resistance and other risk factors in animal models of type 1 and type 2 diabetes, 39 to 47. More specifically, taurine administration has been shown to prevent high glucose-induced microangiopathy, i.e., vascular endothelial cell apoptosis, 48, and in fructose-fed rats, it has been found to restore glucose-metabolizing enzyme activities and improve insulin sensitivity by modifying the post-receptor events of insulin action, 49. The suggestion that nitric oxide, NO, may be implicated in the pathogenesis of diabetes prompted a study to determine whether endogenous NO synthesis or local reactivity to endogenous NO might be impaired in patients with type 1 insulin-dependent diabetes mellitus, 50. The results showed that either NO synthase activity is increased or NO sensitivity is decreased in type 1 patients, a good indication that the L-arginine, NO system is involved in the pathophysiology of diabetes and its sequelae, e.g., diabetic retinopathy. Subsequently, the elevated levels of NO were shown to cause upregulation of the taurine transporter gene and a concomitant increase in taurine uptake in human retinal pigment epithelial cells. 51. Taurine's effect on renal function, 52, particularly as it relates to streptozotocin-induced diabetic animal models, is also noteworthy. As Turkman et al., 1995, have shown, taurine ameliorates diabetic nephropathy by decreasing lipid peroxidation and lessening the accumulation of advanced glycation end products in the kidney, 39. However, whether the findings from animal models of diabetes translate to an effective therapy in the management of diabetes in humans is an open question. In this connection, it is important to note that taurine was shown to reduce insulin secretion by beta cells in vitro, 53. Moreover, contrary to the results from animal experiments, a study of 20 obese human subjects with a genetic predisposition for type 2 diabetes demonstrate that taurine supplementation, 1.5 g for 8 weeks, had no effect on insulin secretion or sensitivity, 54. In short, these findings do not support the view that dietary supplementation with taurine can be used to prevent the development of type 2 diabetes. However, it should be noted that this study was clearly too small and of too short a duration to have any clinical significance. Further experimental and clinical studies will be of importance in evaluating taurine's therapeutic potential in the management of diabetes in humans. 45. Similar issues have clouded the relationship between taurine and epilepsy, although there is little doubt that taurine has anti-epileptic activity in experimental animals. The efficacy of taurine has been demonstrated in both naturally occurring and drug-induced epilepsy in cats, 55, mice, 56, rats, 57, and dogs, 58, and evidence that taurine blocks dentatohippocampal synapses, a locus of importance in epileptogenesis, indicates a specific action in epilepsy. Indeed, preliminary experiments in human epileptic subjects confirm the anticonvulsive effect of taurine, but the effects are not robust, nor are they consistent, 59. This may be because taurine does not readily cross the blood-brain barrier, and several taurine analogues that do are currently under investigation for their therapeutic potential, 60. Taurine in the eye. It has long been known that all ocular tissues, both neural and non-neural, contain taurine, 61, 62, prompting a host of studies to identify its cellular distribution, 63 to 66. Quantitative analysis of whole ocular tissue extracts of the rat eye revealed that taurine was the most abundant amino acid in the retina, vitreous, lens, cornea, iris, and ciliary body, 67. The highest level of taurine was, of course, in the vertebrate retina, and an ingenious experiment involving a judicious selection of normal and diseased mouse retinas enabled Cohen and co-workers, 68, to quantify the distribution of taurine and other amino acids across the layers of retinal cells, Figure 2. Note that in the normal, control, retina, taurine exceeds the concentration of each of the other amino acids by tenfold or more, whereas in the photoreceptorless C3H mouse, its concentration is about one-third of its value in the control retina. Note also that destruction of the inner retina by glutamate has little effect on taurine concentration. It is apparent, therefore, that taurine is highly concentrated in the outermost layers of the vertebrate retina. This is consistent with the findings that animals, e.g., cats, monkeys, man, that do not produce adequate levels of taurine experience severe degenerative changes in their photoreceptors and retinal pigment epithelium, RPE, when deprived of dietary taurine. 36, 69-75. Open in a separate window. Figure 2. Chemical and genetic fractionation of the retina. A. Juxtaposed images of histological sections comparing the retinas of a normal, control, mouse, with one whose inner retina has been damaged by glutamate, and another that was taken from a C3H mouse suffering the loss of the distal retina. B. The concentrations of 5 amino acids in each preparation. The latter values represent the averages from 6 different groups of dark-adapted animals. Modified from Cohen et al., 1973, with the permission of the publishers. The selective distribution of taurine within the retinal laminas, as well as in other tissues, is attributable to the presence of both high and low affinity Na+ and Cl dependent taurine transporters, 66, 76, 77. At the cellular level, the taurine content is determined primarily by the sum of 3 processes: I. Its synthesis from methionine/cysteine, 2. Its active uptake by the taurine transporter, and 3. Its release via a volume-sensitive leak pathway, 78. The principal transport protein is the saturable, high-affinity TAUT transporter, Km equals 18 μm, a member of the neurotransmitter transporter family that includes the transporters for serotonin, creatine, and gamma-aminobutyric acid, GABA, 79. All members of this family have 12 membrane-spanning helices, with the N and C terminal ends exposed to the cytosol, 80. The cytosolic domains contain several highly conserved serine, tyrosine, and threonine residues that provide sites for phosphorylation. In terms of its stoichiometry, the active uptake of 1 molecule of taurine requires 2 to 3 sodium ions and 1 chloride ion. 78, and only guanidinoethyl sulfonate, GES, and other close analogues of taurine, e.g., beta-alanine, GABA, are inhibitors of taurine uptake. Interestingly, both a GABA transporter and a taurine transporter are active at apical membrane vesicles from bovine RPE. They both require Na+ and Cl and exhibit a similar stoichiometry. An analysis of taurine uptake at this site showed that uptake was severely depressed in the presence of GABA, and conversely, GABA uptake was suppressed by the presence of taurine, 81. Depletion of taurine in rats treated with GES leads to a marked and progressive reduction in the amplitude of the electroretinogram, 82, and severe degenerative changes in photoreceptors and the RPE, 83, effects that can be reversed by intravenous infusion of taurine, 84. A precipitous loss of taurine is also seen after genetic disruption of TAUT in mice. In this model, there is severe photoreceptor degeneration 2 to 4 weeks after birth, and this spreads to the inner retinal neurons after 4 weeks, 85. Clearly, endogenous taurine is crucial for preventing retinal neurodegeneration. Findings such as these, although difficult to interpret precisely, add to an appreciation of the importance of taurine in the cell biology of the retina. Taurine and cytoprotection. Photoreceptors are considerably richer in taurine than other retinal neurons, but all retinal cells from the outer and inner nuclear layers to the ganglion cell layer, and seemingly the radial glia, Muller cells, as well, 86, take up taurine from the extracellular milieu, 66, 87-91. Therefore, it is not surprising that depleting endogenous taurine by the genetic knockout of TOT or by blocking the taurine transporter with GES has been shown to cause ganglion cell loss, along with degenerative changes in the distal retina, 75-85. It is apparent that taurine serves a neuroprotective role in ganglion cells, as well as in photoreceptors. On the other hand, it was surprising to learn that the early in vivo experiments on the active transport of taurine through the frog RPE showed the main flux to be in the retina to choroid direction, 92. However, the ship was righted with evidence of bidirectional transport, 93. And the demonstration of taurine transport from the blood to retina direction, 94. In addition, passive diffusion of such a small molecule as taurine would allow it to traverse the plasma membrane of retinal cells without the aid of an active transport mechanism, and there is experimental evidence that both path length and matrix components, collagen and elastic tissue, influence the diffusion of taurine across human and bovine tissues comprised of Bruch's membrane, choroid, 95. Perhaps the most exhaustive body of experimental work on the neuroprotective properties of taurine was performed by Wu and colleagues, 29, 30, 96-98. These innovative studies provide convincing evidence that there are several avenues by which taurine exerts its protective role. Using primary neuronal cultures from the fetal rat brain, these researchers showed that taurine suppresses glutamate-induced toxicity through several pathways. It inhibits calcium influx through L-N and P/Q-type voltage-gated calcium channels. 2. It prevents the downregulation of BCL2 and the upregulation of BAX, the protein products of which otherwise would translocate to the mitochondria and result in the release of the highly toxic cytochrome C, Cyc. 3. It protects neurons from oxidative stress, and 4. It inhibits glutamate-induced calpain activation, thereby preventing the cleavage of BCL2, see also, 99. There is obviously a broad array of mechanisms by which taurine serves its cytoprotective role, but the molecular identity of a taurine-selective receptor remains a mystery. Several studies have implicated a metabotropic GABA binding site as mediating the action of taurine, particularly in the brain regions of the mouse and rat, 100,101, as well as in the mammalian retina, 75. However, the pathway linking the GABA receptor to its physiologic action has yet to be identified, and there is a high level of uncertainty regarding the existence or nature of a taurine-specific receptor, see below. An experimental study. One of the many experiments demonstrated